of the other components here, so kind of zooming down or kind of zooming in a little bit more on the actual structure of the thin filament or actin. So actin is the contractile protein. It's made of G-actin monomers. And so here's what a single G-actin looks like. And G-actin here, the G stands for globular because it's a globular or rounded or individual form of this actin. It's going to have this open binding site for the myosin. We'll talk about how myosin will actually make contact with actin in a bit. But if you look at a single unit of the actin filament, it's made of these repeated G-actin molecules. Now, if we were to line those up, right, kind of like we would pearls on a string, we would form what's called F-actin. And F-actin stands for fibrous or filamentous. And so it's not the globular form of actin for the individual units, it's now the F or fibrous form as we connect those globular molecules together on a string. Now we're going to take two of those F-actin chains, right, or strings, and we're going to intertwine them together in a double helical actin strand. So the way that we see actin, it's really several globular units attached together, and then two of those strands intertwined to form a double helical strand. That is the actin itself. And then there are two other regulatory proteins that are associated with actin that are going to be really important as far as the regulatory components, how actin actually brings about muscle contraction. The first one is tropomyosin. Tropomyosin is this thread-like protein that's running around that helical strand. And then troponin is the protein that has three different binding sites, right? So it's that little pink structure here that actually has three separate units on it. One of those is to attach to the actin. So we can see that first unit actually binding onto the actin strand at the bottom. The second site in the middle is to attach the tropomyosin, which is that thread-like purple protein here. And then the third site, which is bare or exposed, is for the attachment of calcium. And so when calcium binds to troponin, that entire troponin has a configurational shift, and it's going to slide the tropomyosin, which is the thread-like protein, off of the myosin binding sites. So this thread-like protein, which is the tropomyosin, is usually covering up these open areas on the actin strand, which are made for binding myosin, which we identified up here. And so those are typically covered up, preventing myosin from making any contact with the actin. But when calcium comes into play, calcium binds to the exposed site on troponin. That causes the entire troponin molecule to shift. And because one of these sites of troponin has attachment to tropomyosin, that drags tropomyosin away from the binding sites, essentially uncovering or exposing those myosin binding sites such that myosin can now come and make contact. In that way, calcium is sort of the light switch. Calcium is what is turning the contractions on and turning the contractions off by binding onto troponin and causing that configurational change. All right, let's switch gears and look at myosin. So we've talked about how actin looks, the regulatory structures that are kind of exposing the myosin sites on actin. Let's look at the myosin filaments on the other hand. So this is the thick filament. It's created by two filamentous or myosin strands that are intertwined onto each other, kind of like golf clubs, right? So there's two filamentous proteins intertwined together, similar to what we saw with actin. And then the heads of the myosin are sort of curved upward, and they have two binding sites. One is for actin and one is for ATP. So we can already begin to see that myosin has the site which can bind the ATP, which is going to be responsible for energizing this whole process. So the myosin heads can bind the actin, which is going to be important for the actual shortening of the sarcomere. but it can also bind the ATP, which is going to energize that action. So notice at the bottom here how we have several different myosin heads that are sticking up from the thick filament. So while we have been speaking about the thick filament as being myosin, I want you to understand that it's several different myosin that together are forming that thick filament. Same thing is true with actin. We have several different actin units that are together forming the thin filament. All right. Other proteins that are important for muscle contraction is titin. Titin is kind of like an elastic coil, kind of like the Slinkies. I don't know if you guys are, familiar with the Slinky toys. Probably giving away my age at this point, but the elastic sort of coiled nature of titin is what is responsible for allowing that flexibility of the sarcomere. So having this coiled elastic structure attaching to the Z disc on one side and then attaching to the edge of the thick filament on the other side, it's going to give some flexibility when the sarcomere does shorten. So the sarcomere will have the ability to shorten or compress, right? And then that coiled elastic protein called titin will allow for that movement. Okay. Let's go ahead and illustrate this here. Actually, we're going to go over, I'm sorry here, I think I already have it drawn. So let's go over the illustration here instead. Okay, so we've got this illustration that shows the structure of the sarcomere, where it is in comparison to the entire muscle, as well as the different components and regulatory proteins that are associated. So here's our muscle body. This is covered by the epimysium again. Here's our fascicle covered by the perimysium. Here's our muscle fiber or cell, which is covered by the endomysium. And then within that muscle fiber, we're going to pull out one single myofibril. And then we're going to look at one individual unit on the myofibril, which is the sarcomere. Okay, so this is the level at which we're looking at in the entire muscle. So if we take this single sarcomere, it's going to be from one Z disc here or Z line, and I'm using those terms interchangeably. Z disc, Z line, same structure. On the other side, here's another Z line. And then in the center, we have the M line. So the M line is where we have the myosin heads that are sticking out from the center of the sarcomere and out towards where we have the thin filaments. So here are the thick filaments, which are myosin sticking out, trying to make contact with the thin filaments, which are actin, which are sticking out from the Z discs. So the actin filament is in red here. This is sticking towards the center. And then the myosin filaments are shown here with the blue heads sort of sticking out. The green coiled structure is the titin, again, giving that flexibility and elasticity to the shortening of the sarcomere. And then if we think about our band, so the I band is the area of only actin. If you were to cover this up, you would realize that the myosin does not extend into this region, so it's only the lighter staining region of the I band. The H zone is the central region where there's only the myosin filaments. The A band is the outer region where we begin to see some overlap. So notice the thin filaments in red are overlapping with the thick filaments in blue. And so this is the area where we have overlap. That's considered the A band. And then we can consider the interaction between the thin filaments and the thick filaments as a cross bridge. So the cross bridge is really where the myosin heads are sticking up, right? The blue structures here, and they're making contact with the actin for the red filaments sticking out in between. That is considered a cross bridge. And we're going to talk about how that interaction, right, actin to myosin, is what brings about the cross-bridge cycling, which is what brings about the shortening of the sarcomere. Okay. The other thing we want to say here is kind of what's happening. What's the direction of movement? So when the sarcomere shortens by way of actin making contact with myosin, the Z discs are going to be drawn closer and closer together. So we're going to have cross-bridge cycling occurring, which is bringing the sarcomere to be shorter, right, or smaller. In doing so, actin is being dragged towards the center of the sarcomere. So here's actin. It's going to be dragged in this direction. Myosin is going to be moved in this direction. And so the Z discs are going to be moving closer and closer together, which is what shortens the sarcomere. That is going to effectively shorten the myofibril. That's going to shorten the muscle fiber. It's also going to shorten the muscle fascicle, and then we're going to shorten the entire muscle belly. So this is the unit at which we see the muscle shortening, which brings about the overall muscle contraction. Okay. Okay. Any questions on this so far? Any questions? So far, we've gone over the basic structure of muscle, and we're thinking about what are the different levels of the muscle, all the way down to the cellular level. What are these subcellular components, right? Things like lots of calcium storage in the sarcoplasmic reticulum, lots of mitochondria to create lots of ATP, and then the individual units in there, which are the myofibrils. And then we also talked about the units, the repeating units on the myofibril, which are the sarcomeres. So we've kind of set the stage so far to understand how this can be put into place. What are the structures? What are the players in place? And how they can act together to bring about that shortening that we just described. So the shortening is described as the sliding filament model. And the reason that we call this a sliding filament model is because no one has ever seen this happen. Really interesting. The early physiologists and early anatomists and histologists that were studying muscle, no one has ever actually seen muscle contract under the microscope because that is just not feasibly possible. In today's world of more advanced technology and more advanced visualizing techniques, and histological methods, we can probably better visualize muscle contracting. But when we first began studying muscle, we had a model, we had a theory of how muscle contracted based upon those staining patterns that we saw in the relaxed state of muscle, and then the staining patterns that we saw on the contracted state of muscle. But no one has actually ever seen this contractile process take place. So for that reason, we call it a sliding filament model. The cross-bridge cycle is going to be the cyclical movements that are responsible for that shortening of the sarcomere. Essentially, on a macroscopic level, this is how the muscle generates its force or contracts. And then we're going to talk about excitation, contraction, coupling. This is how we use calcium to turn muscle contractions on and off, right? We talked about how calcium and troponin played a regulatory role. And so excitation, contraction, coupling is turning those contractions on and off with the help of calcium. And then we can also look at muscle metabolism. This is how muscles provide the ATP that is essential for driving cross-bridge cycling. So we looked at the structure of myosin having the binding site for ATP. And so ATP can bind to myosin, and this is how we provide the energy to actually drive the muscle contraction. Now, muscle contraction is the shortening of the muscle. It is the thin and thin filaments being overlapped. But here's the thing. Neither of these filaments are actively shortening, right? It's really important to understand. Nothing is shortening. Nothing is, how do we say, contracting on its own, so to speak. What's happening is myosin is going to make contact with actin, and then myosin is going to drag the actin closer to the center of the sarcomere. So the filaments are really sliding past each other, or being dragged past each other rather, instead of actually shortening on their own. The shortening is happening on the level of the muscle. When we think about the sort of zoomed out macroscopic picture, the muscle is shortening, but at the level of the sarcomere and the filaments that are there, none of them are really shortening. They're kind of just sliding past each other. And so this is again how we get that sliding filament model. So in the sarcomere, during contraction, the A band is going to remain the exact same length, right? So here's the A band, the region of overlap, right? Here's the A band. Notice how when the muscle is contracted, the A band is the exact same length as it is when the muscle is relaxed. So up here, the muscle is relaxed. The Z discs are slightly out further, slightly further away from each other, right? And so what's happening is myosin is dragging actin towards the center. Notice how actin is really far away from each other here in the relaxed state, but it's really close together down here in the contracted state. So the proteins themselves are not shortening. Filaments themselves are not shortening. If you look really closely, they're staying the exact same length. What's happening is they're just sliding closer and closer towards each other and further away from their point of origin. So in doing that, because myosin is moving in this direction, actin is moving in this direction, the A band stays the exact same. It's the area of overlap, so that makes sense. The area of overlap would stay the same if the size or length of filaments are staying the exact same. The I band is going to shorten. So here's the I band. It's the area of only thin filament, so this region out here. That is going to shorten because guess what? Myosin is moving further in this direction, actin is moving further in this direction. So if you look at the contracted version of the sarcomere down here, more of the actin has been dragged towards the center, and so the length or the size of the I band is going to get shorter. The H zone is going to shorten as well. We looked at that earlier. As actin is being dragged closer and closer to each other at the center, the area where there's only myosin gets shorter and shorter as actin sort of invades that space in the H zone. The sarcomere on a whole is also going to shorten, again, because we're dragging those actin filaments towards the center. This Z disc is going to get closer to this Z disc, and the entire unit is going to become shorter or smaller. So the only measurement or the only staining region that stays the same is the A band. The A band is the only area that remains the same in the relaxed state of the muscle and the contracted state of the muscle. Everything else gets shorter as the sliding filament happens, as we make the cross bridge. The cross bridge, again, is the contact made between myosin heads and the actin or thin filament. So we make that cross bridge, drag actin in this direction, and then we're going to release that cross bridge and then make another cross bridge, drag actin in that direction again and again and again and again until the entire sarcomere shortens. So notice how not only is that a process of dragging the filaments closer together, but it's also a cyclical process of making and breaking those cross bridges. Okay, looking at the histological view here. So here's our I band, this light pale staining region of only thin filaments. Notice how it's much larger in the relaxed state of the muscle when the sarcomere is nice and stretched. And then it's much smaller in the contracted state of the muscle when the sarcomere is shortened. Notice the so we just talked about the I band. Notice with the location of the Z disc, excuse me. So the Z disc here, right? The Z disc is out here and out here in the relaxed state, and then it's much closer down here in the contracted state. Notice also the location of the H zone. So the H zone is larger the area of only thick filaments or myosin. And then down here in the contracted state, it becomes much smaller. So the I band shortens, the H zone shortens, the Z discs get closer together, the entire sarcomere shortens, but the A band is remaining the exact same. All right. Let's talk a little bit more about the cross-bridge cycle. So cross-bridge cycling is making contact between actin and myosin, dragging the actin towards the center of the sarcomere where we have the M line, breaking that contact, going out, making contact again, dragging it again, breaking contact again and again and again until we have dragged the actin filaments as close to the M line as is possible, which is shortening the entire sarcomere. So the myosin head has to go through some conformational changes back and forth as it makes and breaks these cross bridges. So it's going to be in two different states in order to do that. It's going to be in a high energy form. This is where it has ADP and a phosphate bound to it, an inorganic phosphate. That is where it's going to have a high affinity for actin. It's going to have a high attraction to actin. It's going to really want to reach out and bind to that actin. Alternatively, it's going to be in the low energy form when it has dragged the actin. So after it has dragged the actin towards the M line, it's going to now be in the low energy form. Now it has ATP bound to it. Now it has a low affinity for actin. And here it's not going to be attracted to actin. It's going to want to break that attraction. It's going to have a low affinity for actin, and it's going to want to release the actin in order to break that cross bridge. So in order for this to go from the low energy form back to the high energy form, it's going to require the hydrolysis of that ATP. So to go from the low energy form where it doesn't like actin, it doesn't want to bind, to go to the high energy form where it really wants to bind actin, we're going to have to hydrolyze the ATP that is bound on the myosin site.